Hey guys, so this video is going to be the entirety of what we started off in class on Wednesday and Thursday respectively, I guess. I'm hoping that it's just going to be like a refresher, and most of these terms and methods are things that you are familiar with and have definitely seen before. So here we go. So the father of genetics is known as Gregor Mendel, who started his initial studies in like the mid 1800s. Gregor was a monk, so just proves that science and religion can sometimes work together, who documented just observations he made based on inheritance in pea plants. Now, even though it was the 1800s and we didn't know about DNA and we for sure didn't know about chromosomes, he was able to identify things that are controlled by your DNA and by chromosomes. He was also able to successfully use the scientific method, even though that in itself didn't really exist yet either. So what did Mendel do? Mendel took two true breeding or pure plants and cross-pollinated them. True breeding means that you are either homozygous dominant for one trait or homozygous recessive for the other trait. These two initial plants were his parent plants, or parental generation. It's always signified with this capital P. So he cross-pollinated them, and he collected the seeds and observed the seeds, and then planted them and saw what happened. Well, the first generation, which is the result of these two peas mating, were all purple flowers. So if you think about that, completely purple flower paired with a completely white flower gives you offspring that are all completely purple. So then he said, well, let me see what would happen if I allow this F1 generation to self-pollinate. So he did. He allowed them to self-pollinate, and then he grew those plants and made observations based on what they looked like. So in total, there were 7 different traits that Gregor ended up making observations on for a various amount of time. Not only did he look at things like the color of the flowers, but he also looked at the color of the seeds, the shape of the seeds, the color of the pods, what shape the pods took, where the flowers were located, and the height of the overall plants. So let's look at it again. So in our P generation, in our parental generation, we're taking a true or pure purple flower and crossing it with a pure white flower. The first set of offspring are completely purple. He then let those offspring self-pollinate, and the result, or the F2 generation, were peas that were purple, but also peas that had, so not peas actually, but pea flowers, that were purple, and pea flowers that were also white. So in our F1 generation, 100% purple. In our F2 generation, 75% purple, 25% white. What does that tell us? Well, it has to tell us that our F1 generation aren't pure purple, that there are some white traits somewhere in their genotype, but that's masked by their phenotype. Their phenotype is purple, but their genes still contain the genes from their white parent. If that weren't true, when they self-pollinated, we would have gotten all purple flowers again, instead of this 25% chance, of one of the flowers or some of the flowers being white. So what does it mean? Well, Gregor figured out that traits or characteristics have alternative versions. It's kind of like cookies on the cookie aisle at Walmart or Kroger. If you counted all the different types of Oreos you saw, or all the different types of Chips Ahoy that you saw, you wouldn't just have your standard Chips Ahoy or your standard Oreo. You'd have lots of different varieties. Like, I was in Target the other day and they now have ice cream Chips Ahoy. Like, really? But anyway, but you get what I'm saying. You still have cookies, you just have lots of variety. Just like with these peas, they still have a color for their flowers, but there are 2 options. There are 2 varieties or 2 alternatives. In genetics, those alternatives are called alleles. So allele is a different version of a gene. All right? The correct scientific definition is that these alleles, these different versions of your gene, or these alternate versions of your gene, may vary based on the sequence of their nucleotides. So you're going to have some change between those base letters of A, T, C, and G. But they are found in the same location. By the way, this word locus, which is singular, we use the word loci for plural. It means location. All right, so position on the chromosome. What chromosome it's found on and where exactly on that chromosome. That's what your locus is. Now, let's think about what we know so far. We know that you get a chromosome per parent. You know that all of your characteristics are controlled by those genes on those chromosomes. Okay? So technically, for every characteristic, you, the organism, inherit two alleles. One allele from mom, one allele from dad. Right? This is the whole reason why we're diploid, right? Remember that word that we talked about before? This 2N condition, where for human beings it means that we have 46 chromosomes. We have those 46 because we do get a chromosome from each parent, but every chromosome carries two different alleles, one allele from mom and one allele from dad. Okay, so these are our homologous chromosomes that we've talked about before. It's almost like having two versions of something, like an encyclopedia, for example. There's Encyclopedia Britannica and Encyclopedia Americana. It just depends on which one you have and which one you want to use. So what is the advantage of being diploid? Well, you get two of every allele. So if something goes wrong with one, so if one malfunctions, you may have one that works. And you really only need one. You need one to be expressed. So it's not necessarily a bad thing. It's like having a backup plan. Now, let's go back to what happened when he crossed those first true breeding flowers, the true breeding white and the true breeding purple. The ending result was an offspring or a generation that was completely purple. Like every single flower produced was purple. So why didn't the white show through? Or why didn't we get like a lavender flower instead of like a just plain purple flower? Well, that's something else that Mendel discovered. Some traits mask the presence of other traits. So in the case of our pea flowers, the purple trait and the white trait are separate. They don't blend together. It's either your purple or your white. And in this case, the purple color masked or overcame or suppressed the white color. So that means that our purple color, our gene that gave us that purple color, or our allele that gave us that purple color, is what we call the dominant allele. Now, when you were in freshman year, we probably told you that your dominant allele is your stronger allele. But really what's happening is your dominant allele is your functional gene. It is the gene that is going to make a protein that works or that functions. So that's why you're seeing the purple color, because it's the gene that creates that purple color that's the one that's working. Whereas with your recessive allele, the allele makes a gene that's makes a protein, that's malfunctioning. So you don't see it as much. It's almost like you need two of them to be able to see it, hence the recessive trait. So now that gets us into genotype versus phenotype. Your genotype is the description of your genetic makeup. It's all your A's and T's and C's and G's and the arrangement that they come together in to make that particular trait. Whereas your phenotype is your physical description. It's your physical appearance. It's what you actually look like. So if you're tall or short, with brown eyes or blue eyes, and wavy hair or straight hair or blonde hair or red hair or whatever it is that makes you look the way you look, your genotype is responsible for your phenotype. So if we look at our pea flowers over here in this picture, our dominant allele would have been the purple one. That's why in our F1 generation, the plants all look purple. The white allele is recessive because in the F1 generation, you can't see any white. The phenotype that is expressed is the dominant phenotype. The recessive phenotype is not expressed. Now, geneticists a long time ago figured out that it's easiest to use letters to represent your alleles. And the way it works is you choose one letter to represent both traits. So like, for example, if we were working with this purple color and this white color, we would use the letter P to represent flower color. Now, the alleles come in two forms, either dominant or recessive. So dominant would be represented with a capital letter, in this case, big P, and recessive would be represented with a lowercase letter, so the white one in this case would be little p. Now we don't use P for purple and W for white, like that's just too confusing. We choose one letter and we make that letter apply to both traits. So when you are true breeding, it means that you're going to be homozygous. Okay, both letters are going to look the same. Either they're going to be big or they're going to be little. Because you are purple and purple is our dominant allele, the gene that would represent, the letters that would represent the true breeding purple allele would be big P, big P. Whereas the true breeding white allele would be represented with little p, little p. So instead of drawing like all these pictures and coloring them and all of that, we could use these letters because we know exactly what it would mean. When I look at this, I can tell that this is my true breeding purple flower, and this is my true breeding white flower. When I mix them together, I end up with one big P and one little p. But because purple is dominant, remember, this is the protein that functions. This is the color we're going to see. We're going to see this purple color, so that's why this flower looks purple. But I want you to note that we didn't lose our gene for our white flower. It's still there. It's just masked. It's not being correctly expressed. All right, so here we're still looking at Mendel's initial cross, but this time all this picture shows is us adding in those alleles. The ones on the bottom didn't show up, but anyway. So here again we have true breeding purple, true breeding white. When we cross, we get 100% purple flowers, but note the genotype. Note the fact that those genes are a mixture of one big P and one little p. What we see is purple because purple is dominant, but the p, the little p, which represents white, has not been eradicated. It is still there. So when we let these self-pollinate, we know that we have a 75% chance of getting purple flowers and a 25% chance of getting white flowers. But what do the genotypes of this look like? Well, we could figure that out using a Punnett square. So when we go back and we cross that F1 generation, this is how we would set it up. Now this is a Punnett square. A Punnett square is a tool to help you figure out how these traits are going to move around and align themselves with each other. Now remember, our F1 generation are all big P, little p, because they're the result of crossing a true purple plant with a true white plant. The F2 generation comes out 75% purple, or 75% chance of getting a purple plant, I should say, and a 25% chance of getting a white plant. But let's look at the genotypes. There is a 25% chance that you will get flowers that are big P, big P. There is a 50% chance that you will get flowers that are big P, little p. and there's a 25% chance that you will get flowers that are little p, little p. Now this ratio, or these percentages, are only for genotype. Remember what the genes look like, so this column right here. If we put that in ratio form, it would be a 1 to 2 to 1 ratio. But in terms of phenotype, there are only two options. You're either purple or you're white. There's nothing in the middle. So these plants, this one and that one and that one, would all be purple in terms of phenotype, whereas this one would be white in terms of phenotype. So 75% chance of getting a purple plant versus a 25% chance of getting a white plant. But this time our ratio is 3 to 1. Realize and remember that genotypes and phenotypes more than likely will have different ratios. It just depends on the scenario. All right, so let's use some words. And I mean, these are words you already know, but let's refresh our memories on what are the words we're going to use to describe these conditions of uppercase letters versus lowercase letters. So when the alleles are the same, and when we say same, we mean identical. Okay, in terms of letter and size. So when the alleles are the same, like big P, or little p, we use the word homozygous to represent that, to describe that. But when the alleles are different, and when we say different, we're talking about in terms of size. So like big P, little p, for example, we use the word heterozygous to represent that. Now, if you are big P, then you're also dominant. So the term we use for big P only is homozygous dominant. Whereas if you're little p, you're recessive, but you're still homozygous, so the term becomes homozygous recessive. And again, as we said before, big P, heterozygous. Okay, so it's very possible for two organisms to have the same phenotype and have completely different genotypes. So just because the flower is purple doesn't mean it's homozygous dominant. It can be purple and be heterozygous as well. So you can't tell genotype by observation. You can't tell by just looking at the flower what the genes look like. How would you be able to determine genotype if you don't already know? Is there anything we could do? Well, of course there is. What you could do to help you determine genotype is using a Punnett square in a format that we call a test cross. We breed the dominant phenotype with a homozygous recessive phenotype, and that helps us to identify our unknown allele. All right, so let me clarify that again. To do a test cross, you take the dominant phenotype. Now remember, we don't know anything about genotypes of our flower in question. Like, for example, we're trying to figure out what's the genotype of this flower. This flower could be big P, homozygous dominant, or big P, heterozygous. We don't know which one it is because we can't tell by looking. So what we would do is we would take our dominant phenotype, which is this purple flower, and we would breed it with a homozygous recessive flower, in this case a white flower, because in pea plants the only way you can have a white flower is if you are homozygous recessive. So we know the genotype of this one, but the genotype of this one is unknown. We don't know. We only know its phenotype. So let me show you how it works. So this is how we conduct a test cross. You take the homozygous dominant flower or trait and cross it with the recessive trait. Then you take the heterozygous and you cross it with the recessive. How do we interpret the results? Well, if all of the offspring display the dominant phenotype, which in this case is purple, then the individual in question is going to be homozygous dominant for that trait. But if the offspring display both dominant and recessive phenotypes, so in this case purple and white phenotypes, then the genotype in question is going to be heterozygous. It just depends on what they express. Now, another term that you might see instead of test cross is back cross, but it's the same thing. Okay, so Mendel came up with a couple of rules or laws based on the observations that he made. The first one is called Mendel's law of heredity. It's also known as the law of segregation. During meiosis, the alleles segregate. That just means they separate themselves. This is because our homologous chromosomes separate. We separate mom's chromosomes from dad's chromosomes. Since each allele for a trait is found on one of these chromosomes, and since the chromosomes are being separated into different cells, it means that each allele for a particular trait is going to be in a separate cell or a separate gamete. All right, so if we look at the picture, switch to my laser. Here we have the homologs of mom and dad carrying their different alleles. That's what the colors represent. Through meiosis, the first thing we do is we split those homologs apart. And then again, we split them into sister chromatids. The ending result, remember, would be 4 daughter cells that are all genetically different. So each allele ends up in a different cell. We will have one allele here, another there, another here, and another there. Okay, so this is called the law of segregation. Alleles separate from each other. And it's random. Now, which stage of meiosis creates this? Well, that happens, it lines up in metaphase 1, and then the actual separation happens in anaphase 1. And what's crazy is in the mid-1800s, there was no knowledge of DNA or genes or anything like that. So it's ironic that he came up with these based on just pure observation, and it worked out to be pretty accurate. So there's several types of crosses and Punnett squares that we could utilize to help us work genetic problems. The simplest of which are called monohybrid crosses, and a lot of Mendel's experiments used that form of inheritance. We're talking about inheriting a single trait because mono means one. So 2 examples would be like flower color, which we've been talking about this whole time, and also seed color. We're just crossing for one trait. But we also have a dihybrid cross, and like the name suggests, in a dihybrid cross, we're looking at the inheritance of 2 different traits, or the crossing of 2 different traits. So an example would be like crossing for seed color and seed shape. All right, so let's look at how dihybrid crosses happen, because they are the more difficult of the 2 sometimes to understand. So as usual, we're going to start off with true breeding parents. In this case, we're going to look at true breeding yellow and round peas, because remember it's 2 traits, and true breeding green and wrinkled peas. The yellow and round peas are the dominant condition, whereas the green and the wrinkled peas are the recessive conditions, and we can tell that by the size of the letters, big Y versus little y, and big R versus little r. When we first cross, we're going to end up with an F1 generation that are 100%, or there's a 100% chance that the F1 generation is going to be yellow and round. But seeing as how they came from parents that are both yellow and round and green and wrinkled, it means that this F1 generation are all going to be hybrids. They're going to be heterozygous for both traits, for both yellow color and round, appearance. So we do it again. We let them self-pollinate. And when we self-pollinate, something interesting happens. We get a very specific ratio, a ratio of 9 to 3 to 3 to 1. And this is out of 16, just because of how the alleles align. And if you don't understand how we got the 16, then let me know in class and we can go over this. But the vast majority, of your population, 9/16 of your population, is going to be round and yellow. 3/16 will be green and round. 3/16 will also be yellow and wrinkled, and 1/16 will be green and wrinkled. Now remember, a lot of this is just phenotype. We haven't looked at genotype yet. So what's going on? How is this working? If our genes are on different chromosomes, how do we even get them into gametes? And do they move together or do they move independently? Is it always going to be the big ones connecting and then the small ones? Or do we just mix it all up? Well, hopefully you already know that this is the way it happens and not this way, but let's talk about how we. So let's do some proving. Let's use that Punnett square. If the only way they assorted was based on size, big Y with big R and little y with little r, this would be the result. Well, we know that this is true. We know this is fact, that this 9:3 to 3:1 ratio is correct. And here we have a 3:1 ratio. 3 meaning we have 3 out of 4 that are going to be yellow and round, and only 1 out of 4 that's going to be green and wrinkled. Well, that's not right because this is the correct ratio. Well, what happens if we let the alleles just mix themselves up however? If we do that, we end up with all kinds of craziness. Big Y and big R, but big Y and little r, or little y and big R, or little y and little r. When we let those cross, look, we get our correct ratio. 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 16, yellow and round. 1, 2, 3 out of 16, yellow and wrinkled. 1, 2, 3 out of 16, green and round, and 1 out of 16, green and wrinkled. So this proves that. So that leads us to Mendel's second law. It is called the second law of heredity, or the law of independent assortment. And it says that different genes separate into gametes independently. Like which gene on which gamete you end up with, it's all a random process, okay? And it's because non-homologous chromosomes align independently. And then you have gametes that are produced in equal amounts. You have to make 4 gamete cells, and each gamete or each sex cell has to have DNA in it. It must have chromosomes in it. Now, this is only true under certain conditions. It's true for genes on separate chromosomes or on the same chromosomes but are far enough apart that crossing over happens frequently. There are some genes that are an exception to this rule. Now this one is the one that's sometimes confusing, so if you don't understand, make a note, put a star or highlight it or something, and make sure that we go over this in class. All right, so let's look at the law of independent assortment a little bit more closely. Independent assortment, is going to happen or start to happen in metaphase and then continues through anaphase. The only exception, the only way that this does not become random is if the genes are on the same chromosome and are close together, because when that happens, those genes get inherited as a unit. They get inherited together. And sometimes it just makes that crossover really happens. Like they don't cross over separately. They always cross over together. So we call these genes linked genes. It's almost like there's a chain in between the two of them, and if you take one, you're automatically taking the other. They're a packaged deal. It's kind of like, your spouse and their parents. If you don't love them, sucks to be you because you're getting them anyway. Lucky for me, I happen to love the parents and siblings of my spouse, so I was lucky. Okay, so chromosomally, let's look at the basis of Mendel's laws. There are 2 main ones, independent assortment, and segregation. So if we look at the genetic events all the way through meiosis, all the way through when we form our gametes, and all the way through fertilization into a zygote, we see how those chromosomes are constantly moving and constantly changing. And this is one of the reasons why sexual reproduction is only really the only time that we see an introduction of difference. We see that genetic variation. Okay, so let's review. Law of segregation. That's proved through monohybrid crosses or single trait crosses. Each allele segregates into separate gametes. It's established in metaphase 1 because we separate homologs and then we separate sister chromatids, and then each chromatid ends up in a different cell. Law of independent assortment is proven by dihybrid crosses or more, because yes, there is such a thing as trihybrid crosses even. And it's when genes on separate chromosomes assort into gametes independently. Also established by metaphase 1. The only exception to this general rule is linked genes. And linked genes are just genes that are connected to each other. They're inherited as a unit. If you get one, you get all of them that are all together. Now, why did Mendel choose peas? Well, to be honest, I'm not sure if he really chose peas specifically, at least not initially. I think there are just peas in the garden, and he made some observations, and he decided to pursue it. But pea plants are really good for genetic research. They come in lots of varieties, they have some very distinct heritable features, and they come with good variation. So, like flower color, seed shape, pod shape, all that other stuff. Mendel had extreme control over which plants mated with which. Like, he organized all of the mating processes of these particular pea plants. So there wasn't a whole lot of room for error. Okay, he was the one that cross-pollinated them. Both sets that he worked with had both female and male parts, so they could cross-pollinate with each other, but they could also self-pollinate and self-fertilize. And that's a trait that pea plants have. They can fertilize themselves, so they don't really need another independent pea plant necessarily. Pea plants are good for genetic research. They're simple. Like, genetically, they're really simple. So it's easy for us to interpret what's going on with them. A lot of their characteristics are controlled by a single gene, and that single gene only has 2 alleles. We don't have to worry about like messy linked genes and genetic control of one trait over a bunch of other traits. So it was just easy to work with them. One trait's always going to be completely dominant over another trait. All right, so I hope it wasn't too difficult. Again, make sure you look at it. You make sure you go over whatever you took notes over. And if you have questions, please let me know so that we can address them in class. Let's not pretend that we know what's going on. That doesn't help any of us. Have a great night, guys. I'll see you soon.